Hi everyone. In this video, we are going to be talking about central dogma. All right, so central dogma is all about how to convert the instructions in nucleic acids to make proteins. That's the general idea. So we're going to use the instructions in the nucleic acids to make make proteins. Okay, so generally this will follow the pathway from DNA going to RNA, RNA to proteins. Okay, DNA to RNA is a process known as transcription, and RNA to proteins is a process known as translation. Now, we can also go backwards from RNA back to DNA. This is reverse transcription. Okay, there are many cases where this is possible. Eukaryotes use this for their telomeres. So telomerase is a reverse transcriptase. It uses an RNA template to make DNA. There are also many viruses. They're called retroviruses, and they use a reverse transcriptase to convert RNA back to DNA, and then that DNA can be integrated into the genome of whatever host cell has been infected. So that's our overview. We're going to break this apart into sort of two main sections, transcription and translation. Okay, so let's focus on transcription first. So remember, transcription is about going from DNA to RNA. All right, so we're going to use sort of the same logic as DNA replication, except this time, like I said, we're going to be using RNA. So let's say we have some polymer of, how do I want to do this? I want to make that. Some polymer of a RNA. So this is our RNA polymer. Okay. Notice how the 2' hydroxyl has reemerged. This is not present in DNA. Okay, we're going to use the same exact logic. So we're going to have our incoming 3-phosphate nucleotide, except this time, instead of it being in dNTP, like it was in DNA replication, it is an NTP. So this is our NTP. Same exact logic, where we're going to use the 3' hydroxyl, and we're going to form a bond with this phosphate here and break that phosphoanhydride bond. So just like in DNA replication, we're going to use the energy in the phosphoanhydride bond to make a phosphodiester bond. Okay. So when we write this all out, we convert this over. We have our phosphate, ribose. This was part of our polymer. And now we have attached on our second nucleotide. So we've elongated our RNA polymer by 1 nucleotide. Okay, and then we've also generated that pyrophosphate. That then gets immediately hydrolyzed by pyrophosphatase. Same thing. So hydrolysis to 2 inorganic phosphates. Okay. Remember, key difference is that we're now doing RNA. And because we're polymerizing RNA and not DNA, this should not be a DNA polymerase that catalyzes this reaction, but instead should be an RNA polymerase, otherwise known as RNA pol, RNAP, etc., etc. Okay, so this is our basic reaction. We polymerize this out many, many times, but in order to know which incoming NTP we should use, we look to the complement or the template strand. And in this case, the template strand is DNA. So the other way you might see RNA polymerase referred to is a DNA, that is not a D, DNA dependent RNA polymerase, which, oh gosh, there's an abbreviation for it. I think it's like DRP. Is that it? DDRP? It's there is a way to shorten this down. I forget what it is off the top of my head. If you get curious, you can look it up. The most important thing for us to recognize is if you ever get a terminology like this DNA dependent part is referring to, it uses DNA as a template. And the RNA polymerase part is referring to it polymerizes RNA. Polymerizes or synthesizes RNA. So that means there are RNA-dependent RNA polymerases. There are RNA-dependent DNA polymerases. And then our main DNA polymerase that we discuss in DNA replication is very technically a DNA-dependent DNA polymerase. Okay. So, once again, overarching ideas. The energetics are the same as DNA replication. Okay. Or we should say not replication, let's say polymerization. Oh my gosh, why are you doing that? Oh, that's annoying. Polymerization. Okay. So the energetics are the exact same, but this time we're using the DNA template. Think of this like your master copy. And you're going to be making, right, that RNA copy. Okay. And therefore, whereas in DNA replication, we use dNTPs, remember the main incoming monomer are the NTPs, the nucleoside triphosphates, not dNTPs. Okay, so if we use a DNA template to make an RNA copy, we're going to use the same anti-parallel, and complementary rules that we previously discussed. All right, so if we were to have some sequence, let's write it maybe 5 prime TC GG TA TG CAT, I don't know, 3 prime. Okay, and we wanted to transcribe this. Okay, we're going to use anti-parallel and complementary rules because this is now double-stranded nucleic acids. Now, this is going to be during the transcription period. You will actually form what's called the DNA-RNA hybrid. Okay, but they're still, the two strands are still going to be anti-parallel and complementary. So, 5 prime is going to be paired up with 3 prime. T is going to be paired with A. C is going to be paired with G. G is paired with C. G is paired with C. T is paired with A. And now, this here's DNA, right? Here's our DNA template. In transcription, we're trying to make our RNA transcript. So, an A in DNA is not going to pair with T in RNA because RNA does not contain thymine. So instead, that this A is going to pair with U. Okay. All right, T in the DNA pairs with A pairs with U, G pairs with C pairs with G, A pairs with U, T pairs with A, and then we'll put 5'. I would really, really get in the habit of writing down your polarities, because oftentimes, let's say this is your answer, they will give you one, but it will have these polarities flipped. Okay, so it'll be written 5' on the left, 3' on the right with the same sequence. However, it should be inverted if this is your answer, but the answer choices all have 5' on the left and 3' on the right. So be very hyper aware of your polarities. Okay, so we have this basic idea of transcription now. If I were to give you now a DNA sequence, and once again, for funsies, I don't know, let's say 5' up here, AGTT, AGC, TACC, GAT. I don't know. 3'. Okay, and then we'll give its complement. So this is double-stranded DNA. So A is going to pair with T, G with C, A, T, C, G, A, T, G. Oop, that's not where that G should go. G, G, C, T, A, 5'. Okay. My question now can be, all right, which one of these am I going to use as my template? Up here, I gave you just single-stranded DNA, and I told you this was the template. But here, because I've not given you this information, well, we don't know. Either of these could be the template strand. Remember, RNA polymerase is going to read from 3' to 5', and it's going to polymerize 5 to 3. So if RNA polymerase is moving in this direction, then the template strand is the bottom strand. However, if RNA polymerase is moving in the opposite direction, the template strand, well, remember, it's the one that's going to go 3 to 5 because that's the direction it reads, is now the top strand. Okay, so based off of this information, we cannot figure out which one is the template strand or not. Okay, so that will take us into this first part of the more specific transcription initiation. And we're going to focus on the promoter. Transcription initiation does become a lot more complex, especially in eukaryotes. However, for now, we're really just going to focus in on this promoter. Okay, so the promoter is a sequence in the DNA that you'll see either binds or recruits RNA polymerase to begin transcription. Okay. Now, because it does this, there are two main things that the promoter does. Okay. The first one is it tells RNA polymerase the direction. What's that called? To read the DNA. So if it tells RNA the direction to read the DNA, this means it must be telling RNA or telling RNA polymerase which strand is which. In other words, it will indicates to RNA polymerase which strand is the template. Okay. Now, the second thing it does, let me move this a little further down. Okay. The second thing it does is it tells, we're going to sort of put this in quotes here, it tells RNA polymerase how often to transcribe this segment of DNA. Okay, what I mean by that is whenever RNA polymerase, we're going to make sort of a assumption here. Whenever RNA polymerase touches the promoter and it binds to the promoter, we're going to assume that transcription is going to occur. So if we have two promoters and one of the promoters is really, really easy to bind by RNA polymerase, and one of them is not so easy to bind by RNA polymerase, what's going to happen is the one that more easily binds RNA polymerase will get more transcription because RNA polymerase will be more likely to grab on to that promoter and initiate transcription. So this will be talked about much more in depthly next week with transcription regulation, but I wanted to put it on your guys' radar that the promoter also does this. Okay. So what we can do, maybe we'll take this exact same double-stranded DNA just for time. And maybe, what the heck? I'm going to put the promoter over here. So this here's my promoter. And I'm going to say that the promoter is on this left-hand side of the thing it's trying to transcribe. In other words, RNA polymerase is going to bind at the promoter, and because the promoter's to the left, it's going to start here and it's going to travel to the right. So now if we look, which one's going from the 3' direction to the 5' direction? Well, that is this bottom strand, and so RNA polymerase is going to transcribe this bottom strand, so it is the template, to make the mRNA strand. Okay, so once again, if we do this, we'll put 5' here because they should be anti-parallel. T is going to pair with A, C with G, A with U, A with U, T with A, C with G, G with C, A with U, T with A, C, C, G, A, U, 3'. So this is going to be our RNA transcript. And now what we can do is we can look at the similarities. And if we look between this guy and this guy, notice how they're the exact same sequence, except the T's have been replaced with U's. That is because this strand is the coding strand. Remember, if we have, or I should say, we can try sort of a mathematical thought experiment here. Let's say A and B are two different things. Okay? And let's say we can group all of our letters we're going to make into only two categories. So I'm going to have category 1 and I'm going to have category 2. So I'm going to arbitrarily put A in category 1 and B in category 2. And then I'm going to add in that A does not equal C. So A and B cannot be in the same category, and A and C cannot be in the same category. Well, therefore, I don't know why it's doing that. That's interesting. If A and C can't be in the same category, then C has to be in category 2. So that means B and C must be the same. So if A and B are different and A and C are different, and we can only have two possible orientations, because remember, these are complementary and anti-parallel, so they're paired together, well, that must mean B and C are the same. It's the same logic that's happening over here. If our template is anti-parallel and complementary to our coding strand, and the template strand is also complementary and anti-parallel to the RNA transcript, well, then the coding strand and the RNA transcript should have the same exact code and orientation, except for that the coding strand is DNA, so it will contain T, and the transcript is made out of RNA, so it will contain U. Okay. So that is why we call this the coding strand. It is also called the non-template strand. And then this one, the template strand, is also called the non-coding strand. There's two other. There's the sense, anti-sense, and plus and minus strands. We're not going to get into it. Usually coding, non-coding, template, and non-template are sufficient names for our class. Okay. So to recap, RNA polymerase is going to read 3' to 5' and polymerize 5' to 3'. Okay. So now we can ask ourselves, sort of back here with the tells the RNA polymerase how often to transcribe the segment, and we could ask ourselves, well, how that's possible. Right, so if we sort of zoom in over here, if you guys remember earlier, I had mentioned this has to do with how well the promoter binds RNA polymerase. And this, how well it binds, is based off of the promoter sequence. So as we start to change the sequence of nucleotides that makes up this promoter, we can start to change its affinity or how well it binds RNA polymerase. Okay. All right, so what's going to happen is we have our gene. Where's my draw? There we go. Let's say we have some sequence, like I said, 5', 3'. 5', perfect. We have our promoter. RNA polymerase is going to bind. Pol binds. So here's our RNA polymerase. It's going to bind the promoter and begins transcription. Okay. So that is our initiation. Like I said, there's a lot more. It's a lot more complex. There are initiation factors. There's transcription factors. We're going to learn more about them next week. But basically, transcription factors and initiation factors affect how well RNA polymerase binds to the promoter sequence. So within the already sort of basal level transcription capabilities of the promoter and RNA polymerase based off of their affinities for each other, we can further alter that through regulation using transcription factors. Okay. Now, if we compare this to DNA replication, in order for DNA polymerase to replicate the genome, well, we had to unzip the strands. We had to have a primer. We had to have all these things in order to maintain the very, very high fidelity of DNA replication. However, because we, in transcription, don't need that super high fidelity because our master copy, that DNA is fine, we can be a little bit more lenient and allow RNA polymerase to do it all by itself. So in terms of elongation, which is what we're going to move on to next, this is all done by RNA polymerase. So what's going to happen? Let's do something very, very nice. Let's do something like that, maybe. Cool. That way it's maintained. And then we'll put 5 prime here, 3 prime, 3 prime, 5 prime. Okay, so what's going to happen is this is our DNA, and we can draw in our RNA polymerase around it. So RNA polymerase at the, well, actually, first we should figure out its direction. Let's say it's moving this way. If it's going to move this way, it's going to read our bottom strand from 3 to 5, making the bottom strand our template. So we can label that. So our bottom strand here is our template strand. And we're going to get out of this an mRNA transcript. We're just going to draw it like that for now. There we go. All right, so at the beginning, right here, RNA polymerase is actually going to unwind or separate the 2 strands. So it's going to separate the double-stranded DNA. And then at the end, it needs to put the double-stranded DNA back together. So it's going to re-zip up the DNA. Okay. And so inside, we're going to get that bubble that's going to move along. In front of it, we still get our double strand. And behind it, we still get our double strand. But inside RNA polymerase, we get this little bubble. Okay, so now moving on to our transcript. RNA polymerase, once again, it's going to start on the 3' end, work this way. And so this 3' end is going to correspond to this end in our transcript here, right? It's going to be the oldest part, which makes this the 5' end of our RNA transcript. And this is going to be our 3' end. So as RNA polymerase moves to the right, it's going to keep adding nucleotides onto the 3' end. Okay. All right, so in terms of how this stops, we have a few types, but generally, so we're looking at transcription termination. Generally, this is going to occur at what's called the terminator. Requires a terminator DNA sequence. And then this might have the help of proteins. In bacteria, it is the Rho protein, which is spelled out like that. And it's like a P-looking thingy. I can't draw the Rho. It looks like that. Rho protein may or may not play a role. Okay. This leads to 2 main types, the Rho dependent. So this is termination that requires the Rho protein to terminate transcription, and then Rho independent, which does not require the Rho protein to terminate transcription. Okay. All right. So once we've terminated, what's going to happen is the RNA polymerase falls off DNA, releasing the transcript, releasing the RNA transcript. At this point, both the RNA polymerase and this promoter can be recycled, meaning this RNA polymerase can go attach to another promoter in the genome, or another RNA polymerase can bind this promoter to start transcription again. So this is constantly occurring. Okay. in theory, it could be the same RNA polymerase binding the same promoter again. I don't know what the statistical chances are of that. I would guess it's fairly unlikely, but that's not a detail that would ever be tested in any capacity. Okay. Now, knowing that RNA polymerase and the promoter are just constantly undergoing transcription, when one finishes, it starts again, that is absolutely something you should know. All right. And so see how we said this releases the RNA transcript. Let's take a moment to see, okay, what types of RNA transcripts could we get out of this? So in other words, what are the different products of transcription? And there are many, many, many types. The unifying theme is it's an RNA molecule. Now, past this point, they start to diverge. Okay? So our first type we're going to talk about is our messenger RNA, or mRNA. This is RNA that codes for proteins. In other words, its nucleotide sequence corresponds to a amino acid sequence. Okay? Next, we have the tRNA, or the translator RNA. Transfer RNA? It's getting late when I call it a, what's it called? A translator RNA. It is a transfer RNA. There we go. I know how words work. The transfer RNA is our tRNA. And this is going to be the one that transfers the amino acids into the growing polypeptides. But really, this is the one that's translating. It is the one that it converts our nucleic acid language to the amino acid language. Okay? And we're going to see this a little bit later. Okay, the third one you do need to know is the rRNA, which are the ribosomal RNAs. These play both a structural and catalytic role. For the ribosome. Okay, so all three of these are involved in translation, but in different capacities. The mRNA is what is directly translated. The tRNA is really the thing that's doing the translation. And the rRNA is what is catalyzing the translation. Okay, and forming the overall protein structure, or part of it. So the ribosome is composed of both proteins and RNA. Note that there are many, 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 many other types, including microRNAs, silencer RNAs, small nucleolar RNAs, small nuclear RNAs. What else? It's called sRNA, I think. I'm blanking on it. It's been a while since these are more obscure RNAs. You will come across them in more advanced biology classes. For us, you might get to hear about them. You might not. What you will have to know about are the mRNAs, tRNAs, and rRNAs. Okay. Importantly, the mRNA, like I said, is the only one that's protein coding. The only one. None of the rest of them get translated to proteins. Might they be involved in translation? Yes, but are they translated themselves? And the answer is no. Okay, so speaking of translation, we're now going to talk about that. Remember, translation is all about going from mRNA, the nucleic acid language, to proteins, aka the amino acid language. Okay, so we're really going to be focusing in on the mRNA to protein path. All right, so if we have some what's called a nascent mRNA, which basically means newly synthesized mRNA, something like, I don't know, UGAAGA AUGAA UGGAGU CCAGCAU. I don't know. 3 prime. Okay. In prokaryotes that lack a nucleus, this is actually going to go straight to translation. So this is going to happen to this is going to go straight to translation. Very importantly, translation and transcription occur simultaneously. They are coupled. This is really important. Okay, if we go back to our drawing up here, in prokaryotes, this transcription is occurring in the cytoplasm. Because prokaryotes, and like a bacteria, do not have a nucleus. What that means is there's ribosomes sitting all the way around. Actually, they're going to be much bigger than that, but whatever. There's going to be ribosomes sitting right around, because they're also in the cytoplasm, our newly formed transcription. And so what's going to happen is that there's nothing stopping those ribosomes from immediately grabbing on to the mRNA and starting to translate. So if we were to sort of draw this out, here's our RNA polymerase. Maybe here is our mRNA strand. What we can do is we can draw in that ribosome. So the ribosome is going to bind, and once we get to the initiation and elongation in a little bit, this will make a lot of sense. It will bind to its ribosomal binding site and start to move forward. And so you can get what's called a polysome, where you have multiple ribosomes on the same mRNA transcript while that transcript is being formed on the 3' end. It's very, very, very interesting. Okay? So TLDR, transcription and I just dropped my pen. Transcription and translation are coupled in prokaryotes due to the lack of the nucleus. Right? That nucleus is going to separate what's happening inside from outside. And so if we don't have that, we don't have that separation. So there's nothing stopping the translation machinery from initiating translation while the transcription is occurring. There are, fun fact, ways that some bacteria get around this. You will learn them in more detail in your microbiology classes, but there are ways sort of around this. But in general, the ribosomes will just immediately bind and start translating. All right, in eukaryotes, we get time. The reason we get time is because of the nucleus. So, just like in prokaryotes, translation is going to occur in the cytoplasm. However, transcription is going to occur in the nucleus. So the nuclear envelope, aka the nuclear membrane, prevents immediate ribosome access to our nascent mRNA. Okay, so what this does is this provides a what's it called? Like a physical barrier to decouple or separate transcription and translation. So they no longer occur simultaneously, and therefore we now have time to start to mess with our mRNA transcript. So instead of going straight to translation, sometimes, or for a lot of genes, like just literally not even waiting for transcription to be finished, in eukaryotes we have to wait, and we get time to make these processing. So our pre-mRNA, which is produced by transcription, is going to go to our post-transcriptional processing. And this occurs, once again, still in the nucleus. Okay, there are three main things that happen. The first is the addition of the 5' 7-methyl G cap. Essentially what happens is when the mRNA strand is somewhere around like 20-ish nucleotides long, so while it's still getting transcribed, an enzyme comes by and adds on this modified guanine onto the 5' end. Okay. The next thing is the addition of the 3' poly A tail. And this quite literally just means you take the 3' end of your transcript once it is finished transcribing whatever you were meant to transcribe, so finishing off at that terminator, and you just add like, I don't know, somewhere between like 50 and a couple hundred adenines. Just add a bunch to the end. So what's going to happen is both of these are helpful or important in order for the transcript to leave the nucleus. Okay? The poly A tail as well very much helps with stability. Okay, so both of these are going to help leave the nucleus, help the mRNA, I should say, leave the nucleus. This ensures that all our processing is done. So if these are not completed, then that mRNA transcript is not been processed and therefore can't leave the nucleus. Okay, more specifically, or in addition to this, are the methyl G cap will be important for translation initiation in eukaryotes. And the poly A tail is important for maintaining or increasing the mRNA stability. So in other words, it helps resist degradation. As the poly A tail shrinks over time, it becomes less and less stable until it gets fully degraded. Okay, finally, we get splicing. Okay, so let's suppose we have some basic mRNA transcript, and it's composed into sections. Eukaryotic, what we're going to get into this idea of genes, which I wanted to define more formally later, but just think of a gene as like a segment of a DNA that encodes for something. Eukaryotic genes are discontinuous, meaning that there is non-coding DNA in the middle of the coding sequence. So if we were to draw this out, we have sort of, like I said, two components. We have the exons and we have the introns. Okay, so on the 5' end, we're going to find our first exon, denoted by E1. It will then immediately be attached to, so these are essentially just segments of our mRNA. It will be attached to intron 1. Oh my gosh, it does auto. That's cool. Okay, then E1 is going to be attached to, I1 is going to be attached to E2. E2 is going to be maybe attached to I2. And then maybe, just for fun, we'll throw on one more exon at the end here. Exon 3, and then we'll get our 3' end. So with splicing, using the enzyme or the complex, the spliceosome, yeah, there we go. That's how to spell it. What's going to happen is step 1 is remove the introns. So what we're going to end up getting is a 5' with just our exons added together. So this can be kind of confusing. A lot of students see intron and think it gets kept in, but in reality, we're looking for what gets expressed or what gets shown. And those are the exons. So think exon is expressed. The introns are cut out. Okay, so this is removing the introns. Step 2, which technically they'll occur at the same time, but I'm just going to break it down into steps. Doesn't necessarily have to occur, but often does, which is alternative splicing. Which is basically removing exons to generate different proteins. So here we have one possible mRNA transcript that can be produced. If we were to get rid of E2, we could actually make a completely new transcript that is just E1 attached to E3. Now we're going to end up with a completely, or not completely, but a fairly different protein because it's lacking that complete middle section, right? And remember, what we're going to get to is this idea that we can convert this mRNA into an amino acid sequence. Okay, maybe instead of having all three, or maybe just E1 and E2, maybe what we do is we keep E1 and we keep E2 and we get rid of E3. Maybe that's what happens. Okay? All of these help increase the amount of diversity available to the cell in terms of the proteins it can create. Okay? So this is our alternative splicing. All right. So once we have whatever our final transcript is, this will be called our mature mRNA. And this is ready to go. Actually, we'll say ready to leave the nucleus and go to translation. Remember, translation is going to occur in our cytoplasm. So we're going to meet up with our bacteria here in terms of the translation process. So like I said, we'll first start off with translation initiation. In eukaryotes, what's it called? There's two main types. There's the cap-dependent translation initiation, where, like I said up here, the ribosome will actually bind to the modified cap, and there's also cap independent, which there's an internal sequence. Okay, we mostly focus on prokaryotes in this class, but as always, defer to what your specific professor tells you in lecture. For the purposes of this video, we're going to focus more so on the bacterial one, which uses an internal RNA sequence. Okay, so what's going to happen is the ribosome will bind the ribosomal binding site in the 5' UTR, which stands for untranslated region of the mRNA. Okay. In bacteria, this is called the specific sequence. Oh gosh, let's see if I can remember how to spell it. Is called the Shine Dalgarno. I think that's how to spell it. The Shine Dalgarno sequence. Okay. So if we were to sort of draw this out, we have a 5', our end, then within the untranslated region, so this is before translation begins, we are going to have our ribosomal binding site. And then finally, we're going to have our translation start. Going to have some amount of our protein being translated, and then we're going to reach our translation stop. And then on the end of this is our 3' UTR. So this is our 3' UTR. Here is our come on, there we go. The 5' UTR. As you learn more and more, you will discover how these UTRs are actually very, very important for translation regulation. We're not going to talk about it so much in our discussion. But these are very important later on for getting the correct shape. Now, up here when I said the ribosome, it's actually not the entire ribosome. It's truly the small subunit. So the ribosome is composed of a small subunit that is made of protein, a large subunit, also protein, and various structural and catalytic rRNAs. Okay, so what's going to happen is our small subunit is going to bind at the ribosomal binding site. Hold on, let me finish writing this. Maybe we'll just say that continues on elsewhere because we don't need to focus on that for now. So our small subunit, so this is the small subunit, the circle down here, is going to bind to our ribosomal binding site, and it's going to start to move towards the 3' end. Note that this is moving from 5 to 3. In all other cases, our templates are being read from 3 to 5. For translation, the template, which is the mRNA, is being read from 5 to 3. Okay. All right. Once it gets to this translation start, it's actually going to stop. And we can finally give a name to this translation start, and it is specifically the start codon. referenced by AUG. So the small subunit is going to stop at the AUG. Let's write that down. Small subunit stops at AUG. Okay? This here is the start codon. And a codon is a set of 3 nucleotides that correspond to an amino acid. Okay. So this start codon AUG is going to, in addition to serving as our start of translation, it will also encode the first amino acid in our chain, methionine. All subsequent AUGs, they don't serve as start codons, but instead serve as just a codon for methionine. You do need to memorize that AUG is the start codon. Okay, so once it binds here and stops, it's going to wait until the tRNA that interacts with this codon binds. Okay. So let's see if I can not butcher what a tRNA looks like. Let's see. So they sort of look like that. And this tRNA, hold on. Got to make sure I'm drawing this right. I think that's correct. All right, so tRNA is made out of RNA, which means it's going to have base pairs, which means it can base pair with our mRNA sequence. So at this bottom here, instead of just saying it's a random sequence, we can explicitly write out that it's going to be UAC. And then we can finish off the rest of the tRNA. If we write the polarities, this is the 5' end. And then on the 3' end, we're going to write out 3 nucleotides just because I think they're important. You likely don't need to memorize this, but I think it's good to know. On the 3' end, there are 3 nucleotides that are conserved. It is a cytosine, and then an adenosine on this 3' end. Attached to this last A is our amino acid. Okay, so this here is an amino acyl tRNA. An amino acyl is an amino acid where it's a functional group, so it's attached to something else. Not as like an amino acid part of the chain, but like the amino acid itself is attached to something else through usually its C terminus. And in this case, it's attached to a tRNA. So this is the, in this case, actually, I shouldn't just say amino acid. I should say methionine. This here is methionine bound to the tRNA that is for methionine. So tRNA subscript met indicates that this is the tRNA with the anticodon that binds AUG, and that tRNA is what's known as charged. In other words, it is bound to its amino acid. So this here would be a charged tRNA, and we'll go over tRNA charging in a second. All right, so once this binds, let me copy this down again. Actually, I didn't need to do that. Oh, well. I'm going to simplify our drawing just a little bit. I'm going to replace the more complex tRNA with just a little box with a M sticking off it because that's the 1-digit code for methionine. Okay, once our tRNA binds, we get the condensing down of the large subunit. So the large subunit sort of looks like that. It's going to condense down, and it has 3 primary sites. We have the A site, the P site, and the E site. Notice how the first amino acid, the first tRNA, that is in fact not a T, first tRNA enters the ribosome, specifically the large subunit, enters the ribosome through or via the P site. All other tRNAs will enter via the A site. Okay, so first one will go through the P site, all the rest will come through the A. Once we have our small subunit, our mRNA, our initiator tRNA, and our large subunit, this is our completed ribosome complex, and we can begin elongation. Okay. So in terms of elongation, there are some energetics here. Some of the professors cover it, some of them don't, so I will leave that to lecture. The general idea is that for the ribosome to first off attach, it requires a bunch of elongation initiation factors. In eukaryotes, it's primarily the EI4 family. So EI4E is very, very involved. EI4F is very, very involved with getting translation initiated. There's also the EITUs. There are quite a few initiation factors. So I will leave it up to the specific professor of whether or not you need to memorize all of those. In order for all of that to work, though, there has to be sort of an energy input, and the primary energy input for translation is GTP. So whenever the ribosome moves or these initiation factors do their job, it usually comes at the cost of the hydrolysis of ATP. Hydrolysis of GTP. Sorry, it's late. GTP is the major source of energy for the ribosomal function. Okay? And all like the condensing down and the movement. All right, so zooming in on the elongation now. So we can come back here. We can look at translation, elongation. Okay, we can take this guy as our, what's it called? Our sort of scaffold. And I actually need to pull up a codon chart because I only have the start and stop codon memorized. I don't have all 64 codons. But luckily on an exam, like I said, you'll need to memorize the start codon. If you need any other codons, the codon table will be provided to you. All right, codon chart. So let's suppose that right next door, are you going to delete? Thank you. Right next door was AAA. Okay, what's going to happen is our next tRNA is going to come in through the A site. And AAA corresponds to lysine, which is amino acid K. what's going to happen is this bond here between the methionine and its tRNA is going to be broken, and it's going to be moved to attach to this K here. So if we drew out that a little bit, cool. So if we drew this, sort of going step by step. Oop, Control C, there we go. Why are you pasting all the way down there? I don't know. So going through, that bond is going to break. And now, hello, thank you. This program is so goofy to draw on. Now what we get is our methionine is going to be attached to our K, which is going to be attached to its tRNA. And the methionine tRNA now won't have anything inside of it. Immediately following this, the ribosome is going to move, which is going to require GTP hydrolysis. And it's going to move by one codon. Remember, a codon is a 3 groupings of the nucleotides. So if we drew this out now, we have our 5 prime, AUG, and then AAA. And then maybe we'll add in UCU next. 3 prime. Okay, our small subunit is now going to be mostly centered around the middle codon. We can draw in our tRNAs. Now the tRNAs didn't move, but the ribosome complex shifted over. So now methionine's tRNA is going to be with its AUG still. The one that is attached to K and M, so our growing polypeptide chain, is still going to be there, but now it's going to be in the P site. And in fact, we can draw in our ribosome now. There we go. So here's our E, P, and A. And now we have an open A site for our next tRNA to come in. So this tRNA that's going to enter corresponds, what did I choose? UCU, that corresponds to serine. And now we have a repeat of what we established earlier, where this bond is going to be broken right here. Oop, no, this bond. That bond's going to be broken. The M and K are going to be attached to the S, and then everything's going to shift over. So this is just going to keep on repeating until we get to the stop. Now, once these uncharged tRNAs are in the E site, they get ejected out. Okay, so this is our ejection. And this basically just keeps tRNAs cycling on through. All right, this happens until we get to a stop codon. Okay, so this here is translation, termination. So at these stop codons, once again, you don't need to memorize them, but if you're curious, the stop codons are UGA, UGA, UAA, and UAG. Okay. When the ribosome encounters these, a release factor protein enters the A site, causing the ribosome to disassemble. Disassemble. I think I spelled that correctly. Hopefully. So, importantly, these codons do not code for an amino acid. So when we're writing our amino acid sequence, we should not include a stop at the end because these do not encode for an amino acid. All right? Okay. So now I want to focus a little bit more on these tRNAs because this is going to sort of give this reaction a bit more sense and going to lead us into this idea that even though we're reading 5 to 3, we are going to make our amino acids N to C. So we're going to focus on tRNA charging for a little bit. The enzyme that does this are the amino acyl tRNA synthetases. In the overall reaction they catalyze is some amino acid. Hello? Cool. All right, sure. I don't know what this thing is doing. So the reaction is ATP plus the amino acid plus the tRNA that corresponds to that amino acid ends up going to AMP plus 2 inorganic phosphates plus the amino acid attached to its tRNA that corresponds to that amino acid. And this is using, like I said, the amino acyl tRNA synthetase. This is the overall reaction it catalyzes. Okay, you might get to see the mechanism a little bit more in detail, but this is the overall. You're going to use ATP to connect the tRNA to its amino acid. Okay, so remember how up here I said it was a CCA? Okay, so if we were to draw sort of the very end of our tRNA, so the tRNA is going to have a, what's it called? Can you not? This thing's so annoying. So we have a cytosine, and then an adenosine, or an adenine, and then we also have our 2' OHs, which is good to draw in. And then over here, we're going to have our 3' and our 2' OHs. Okay, and let's say for fun, this is for, let's just do methionine. Why not? So this is the tRNA for methionine, and we're looking at its 3' end. Okay, if we draw out methionine, so it's 2 CH2 groups followed by an SCH3. What's going to happen is through the use of ATP, we're going to connect this oxygen to the carbonyl carbon. So like I said, this is going to expend ATP, and it actually uses so much that it goes not to ADP, but it goes to AMP and to inorganic phosphates. Okay, when this happens, for just the sake of time, I'm going to copy this. Cool. So now over here, let's do it like this. Come on. Really? Okay. I don't. This thing's busy. Why is it so delayed? Oh my gosh. Come on, program. You can do it. Okay. We'll do this just for the sake of space. We'll do that. There we go. Just writing it out slightly different, but the same thing. So now we have made this bond with our tRNA. So this is now our charged methionine tRNA that corresponds to the undo. I don't know why it's lagging so much. I've never had this problem. You can do it. Come on. I don't get it. Okay. I guess the undo is out. There we go. This establishes our charged tRNA. Now, if we get multiple of these, so I'm going to sort of condense this down to this idea of, let's say, our methionine. Actually, let's do it like this. Let's draw out the amino acid. Again, so CH2, 2CH, oh, it should be an SCH3. We'll say O to its tRNA for met. And we're going to bring in, which one did I choose? Let's just follow exactly what I did up here. Potassium. Okay. Or not potassium. Ooh, lysine. So now we bring in the one for lysine. Just 4 CH2s and then an NH2. tRNA for lysine. There we go. So what's going to happen, remember how we said the bond between the tRNA, the methionine, and its tRNA is broken? That's exactly what's going to happen. We're going to break that bond. And well, if you guys remember back to week 2, if we break this bond, it's going to then attach to that nitrogen there. So we're going to end up getting, let's see if I can maybe, see if I can move this over a little bit. That way I can draw it going straight down. Perfect. Okay, so we're going to end up getting is our H2N. For our methionine. And now it's going to be attached to our nitrogen. I have to move this over a little bit. Otherwise I'm not going to have enough room. 2, 4, and H2. There we go. And then this is going to still be attached to its tRNA for lysine. And then we also are going to produce that uncharged tRNA met. Okay, so what we saw here is our N-terminal, or the first one that we laid down, the methionine, its N-terminus is retained. So this tells us that the N-terminal amino acid is the first included in our what's it called? Peptide synthesis. In other words, the mRNA is read 5' to 3', and the protein is synthesized from the N terminus to the C terminus. Okay. Last thing to include is this catalysis here is technically by the, well, we say it's by the ribosome, but truly it's the catalytic rRNA in the large subunit that does the catalysis. It's interesting, but sort of a minor point. Okay. All right, so we've basically gone through all the molecular mechanisms, and so our goal now is to put this all together. So we're going to do a practice problem. And our goal is to be able to translate the given mRNA sequence. So let's say I give you 5', G, U, A, U, G. No, I don't want to do that. Oh, come on. Cool. There we go. Now I added my AUG. We'll just keep going. Sorry, I probably should have said this aloud. My bad. Oop, not T. Whoops. There we go. Let's say this is our polypeptide. Okay, so we have A, U, A, A, G, U, A, A, U, G, A, U, G, G, A, C, C, A, C, G, U, A, C, U, A, G, A, G, A, C, C, U, C, C. That is our mRNA sequence. Okay, if we are going to translate this, we need to put ourselves in the sort of headspace of, oop, I just said that way too loudly into my microphone. My bad. Hopefully I didn't just peak there. Whoops. We need to put ourselves into the mindset of a ribosome. So what the ribosome is going to do, right, it's going to bind somewhere on the 5' end, and it's going to start scanning until it gets to the AUG. And my AUG is right here. Okay? Actually, my first AUG, sorry. Where's my first AUG? I think it was earlier. I'll choose a different color too. My first AUG is right here. My apologies. Notice that it doesn't have to be 3 from the end. This is because the start codon, in addition to serving as the start of translation, it also establishes what the reading frame is. And the reading frame is which group of 3 amino acids, or 3 nucleotides is read as 1 amino acid. In other words, because this is our start codon, these 3 are read as a codon instead of it being AAU. Okay. So, why? What the? Okay, sure. So, what I like to do is I scan until I can find my AUG, and then from there I write or underline what my codons are, as this allows me to better keep track of them. And I do this until I encounter a stop codon. And here's my first stop codon, UAG. So this is going to be the end. And I'm not going to write anything here because this is, like I said, the stop codon. It doesn't code for anything. So returning back to here, looking at my codon chart, I know that AUG codes for methionine, and I'm going to write its 3-digit code and then a dash. AAA codes for lysine. UGG codes for tryptophan. ACC, where's ACC? Codes for, was that threonine? Yes, threonine. ACG also codes for threonine. And then UAC encodes for tyrosine. So this here would be my answer. Please work. Why is this program deciding to die on me now? Okay, this guy is my final answer. Note that it does not include stop at the end. We do not write stop. Okay, this is my final answer. All right, so if we were to now put this all together into a gene, okay, well, starting on the sort of, I guess, on the beginning side of things, we need to write our promoter. Okay, so the very first thing we're going to come across in our gene is the promoter. Right, we are essentially trying to put together all of transcription and translation into one and find this information in our DNA sequence, in our gene. Okay, and remember, a gene is a sequence of the DNA that codes for something. If it codes for a protein, it's a protein coding gene. If it's a gene that encodes for something else, it'll be whatever it encodes for. So like, all the tRNAs have their own genes, but they are not protein coding. They are the genes for tRNA. Okay. All right, so after the promoter, we're going to come across the transcription start site. This is denoted by +1. This is the first nucleotide read by RNA polymerase. This is our transcription start site. Gosh darn it. Okay, after the transcription start site, we're going to have our ribosomal binding site. After that, oh, darn it. This program is really, really annoying me. Anyways, after that, we're going to have our protein coding sequence. Can this move down maybe a little bit? Cool. This is going to start with our start codon. Okay, so remember the beginning is denoted by that start codon. Now, what we want to do is find our coding strand, and I see this is my 5' end, so 5' to 3', this is going to be my coding strand. So here I'm going to find my start codon, but remember this is DNA, so this is going to be an ATG. Okay, in the template strand, it's going to be a TAC. This now, like I said, is our protein coding sequence. Oh my gosh. Fine. Sequence. And at the very, very end of it, we are going to find our stop codon. So let's use UAA. So we'll have TAA here. And that'll correspond to ATT in the template strand. Okay, and then finally at the end of this, we will have our terminator. So this will be our transcription stop. Okay. So transcription start and transcription stop are going to be at the ends. The only thing outside of that is going to be the promoter. If we think about why that is the case, well, we need the ribosomal binding site, the start and the stop codons to all be in our mRNA transcript. And we're only going to get our mRNA transcript from what is between our start and our stop sites. So if it is outside the start and the stop sites for transcription, it will not be included in our mRNA transcript. Okay, so while it's very, very easy to confuse the transcription and translation machinery and all the sequences, you do have to be aware of that all the translation sequences that are necessary in the mRNA, they need to be present in the gene in order to make it to that mRNA. Okay? So the ribosomal binding site has to be first before the start codon. That would be like trying to drive without having your wheels on the ground. It's kind of hard. Same thing here. The ribosome has to bind to the mRNA first before it can start translating. And all of that needs to be within the bounds of our transcription start and our stop. Okay. In terms of these numbers, like I said, the +1 is the first nucleotide read. Anything to this direction are negative values. So this would be like -10, -35. Those are common sequences. And this way are going to be positive values. So maybe +10, +30, stuff like that. Okay. And then maybe we can write the other ends, 3' and 5' here. Okay. In terms of eukaryotes, in addition to these protein coding sequences, right, you might have introns. So maybe in the middle here will be an intron as we diagrammed out above. So remember, bacterial and usually archaea, so the prokaryotes, they generally have continuous genes. Eukaryotes generally have discontinuous. All right, hopefully I'm not forgetting anything. The last thing to talk about is the mutations. So what a mutation is a change in the nucleotide sequence that is undetectable. What I mean by this is some mistakes, like let's say DNA polymerase accidentally lays down a T when the template said a G. That would be an error in replication, and that can be detected because the hydrogen bonds don't match up, right? For our base pairing, they should only match up with their base pair, right? So C is with C is with G and A is with T. So if, what did I say originally? A C was paired with a T, I think that was the example I gave. Anyways, what's the role with that? If a C was paired with a T, that's non-correct pairing. DNA polymerase detects that, removes it, and replaces it. Okay. So let's give some maybe basic example. A, C, G, A, T, A, T, T, G. I don't know. 3'. And then that becomes T, G, G, C, T, A, C, 5'. Cool. So now what I'm going to do is I'm going to change this sequence. Okay, so I'm going to mutate it. So we're going to take our A, C, C, G, and maybe I'm going to change this A to another G. T, A, T, T, G. But I'm only going to do it for one strand. So I'm going to have T, G, G, C, T, A, T, A, C. So notice how these two are non-correct base pairing. This could be detected. This would be an error in something like replication. However, if this does not get detected, then what's going to happen is the cell is going to replicate, right? So after replication, each of these are going to be used as their own template, right? Remember the semi-conservative model. So each of these will be a template. So if we do that, come on. Oop, there we go. Well, now we have our new 3 prime, A with T, G, G, C, C. So this is correct base pairing now. A, T, A, C, 5 prime. Okay. And then over here, this guy will also be used. So A, C, C, G, A, T, A, T, T, G. What the heck? Oh, I see. Sorry, I totally flipped this one. My apologies. There we go. Five prime, 3 prime. There we go. Okay, now that I fixed that, as we can see here, this side is totally fine. It's our original DNA sequence. Why did you do that? What the? OneNote's really pissing me off lately. This is fine. Technically speaking, so is this. That is correct base pairing. However, it is not the same as... Oh, that's to the wrong sequence. It is not the same as what we originally had, right? If we zoom out, our original sequence had an A there, and now we have a C. Or we had a T in the 3 prime strand with the 3 prime end on the left, and we have a G there. This could have been detectable by the cell. This is undetectable. There's no way for the cell to have known that that was that change because it is structurally fine. This here is a mutation. This here is an error. This is not a mutation. Error, mutation. Okay. So in this case, all we did is we changed one to another. This is called a point mutation. Okay. Within that, there's multiple different types. Okay. But generally, for point mutations, it's going to affect one single nucleotide. Okay. We can also classify the, what's it called? The mutations by what they do to the amino acid sequence. So the first one is the nonsense mutation. And the one I like to use is UGU because you can basically make all the mutations from it. It's kind of cool. So what a nonsense mutation, well, nonsense mean it does not code for an amino acid. So this is a premature stop. So the example is if we had a UGU and then it were changed to UGA, right? This is in the mRNA, which means the coding strand is going to be a TGT gets mutated to a TGA. So this is the coding strand. All of a sudden, what went from having a cysteine was now a stop. So where it should have been a cysteine and the protein continues on, we now have a premature stop and the protein's going to stop there. This is going to be truncated. Okay. The next thing that can happen is a missense. So it is still coding, but it's incorrectly coding. So what this means is we've changed the amino acid to another. So one amino acid changed to another. So again, UGU is the cool example. So in the mRNA, we can use UGU. And we can change UGU to say UGG. UGG. Once again, in the coding strand, this would be TGT gets converted to TGG. Okay. So remember, the mutation is happening in the coding strand, and the mRNA is getting that mutation during transcription. Okay. So these mutations are going to result in a cysteine now being encoded as a tryptophan. So where we originally had cysteine, we now have a tryptophan in that amino acid sequence. Okay, the next is a silent mutation. You might also hear this referred to as a synonymous mutation. Okay, this is where there's no change in the amino acid. Okay, so once again, our mRNA is UGU, and it's going to become UGC. This is coming from our coding strand, where we had a mutation of TGT becoming TGC. Using the codon chart, this was originally cysteine, and it is now also cysteine. So even though the sequence has changed, the amino acids haven't changed. This is possible due to what's called the degeneracy or redundancy in the genetic code. Okay, so we can also use this time to talk about why it's a 3-digit codon. There are about 20 essential amino acids, and you need one of them to be a start. So you need 20 ways to differentiate between the different amino acids. Well, we have 4 bases we can use, right? U, G, and I guess we'll say, we'll go with mRNA. We'll say U, G, C, and A. So if we have one position corresponding to one amino acid, one nucleotide, one amino acid, well, then we have 4 possible combinations, right? So if we have one spot, we have an A. I need to give myself more room. So for our one spot, we can have an A, a G, a C, or a U. Well, that only gives us 4 possible combinations, and we need 20. So let's try adding in a second spot. Once again, this can be an A, G, C, or U. The total number of combinations is 4 that can be here and 4 here. And so we could have an AA, an AG, an AC, an AU. We can have a GA, a GG, GC, GU. We have a CA, CG, CC, CU, or a UA, UG, UC, UU. Okay. That came out to, what is it? 2 to the 4, which is 16. Did I say 16? Yeah. 16 possible combinations. 4 times 4, which is equal to 16, but that's not, still less than 20. So we need to add one more. So again, 4 possible combinations for this last one, but now this adds in a 4th. So this was just these 2. When we add in all 3, we now multiply 16 by 4, and so we get a total amount of 64. So now we have more than enough to accommodate our 20 amino acids, and it gives it so that some of these guys have some room to movement. So if we look at like leucine on the codon chart, you will see that it has a ton. It has 6 different codons that all encode for leucine. Okay. The reason why that's the case is it is such an important amino acid for protein folding. It's very, very commonly found in the hydrophobic domains, which is why it has so many codons. Okay, so yes, that is why it's a 3-digit, a triplet for the codons, and not like a doublet, a singlet, doublet, quartet. There we go. Anything like that. All right, so back to our mutations. We have one more, and this is a frame shift mutation. And this it does exactly what it says. It shifts the reading frame. So imagine we had, let's grab this sequence here actually. Let's copy this because we can use it to highlight what a frame shift will do. This was our original answer, yeah. Now what we're going to do is we're going to add in an amino acid. Let's say, or sorry, not an amino acid. Oh gosh. We're going to add in another nucleotide right here. And let's say it is, let's go with another, I don't know which one to choose. Let's say a C. So now, once again, we go find our start codon. And the first two are totally fine. Right? We had our AUG, our AAA, but now our third codon is a UCG where originally, why did it? I don't understand why this program is really, really hating me today. Jeez Louise. Okay, back to this. Our third codon was a UGG, and now it's a UCG. Next is a GAC, CAC, GUA, CUA, GAA, GAC, CUC, and actually we never encountered a stop codon. So this would keep going until it fell off the 3' end. And now if we find the code for this, this would be MET, lyse, then UCG is serine, GAC is aspartic acid, CAC is histidine, GUA is valine, CUA is leucine, GAA is glutamic acid, GAC is aspartic acid, and CUC is leucine. So just by adding one nucleotide here, we inserted it, we caused this frame shift mutation. And this will happen if you add or subtract nucleotides in non-groups of 3. So if you were to add or take away 1, 2, 4, 5, 7, 9, that sort of style, or sorry, not 7, 9, 7, 8, you would end up with a frame shift. If you remove 3 nucleotides, well, you've just removed 1 codon. So while you will shorten it by 1 amino acid, you will not cause a frame shift. Okay. All right. This ended up being over 100 minutes long. But like I said, there's a lot to cover. There's a lot of detail here. And it's very, very, very easy to get confused. So hopefully you guys get a chance to really go through, pause this video as you went, take lots of good notes. As always, if you have any questions, please let me know. I'm happy to answer them. All right. I wish you guys the best with your studies, and I will see you guys next time.